So, hello everyone, welcome back to yet another episode in my YouTube channel. Today I am going to talk about principal component analysis, which in short also known as PCA. If you are wondering about how to perform principal component analysis in order to study genetic diversity, then this video is for you. In this video, I will focus mainly on the syntax or coding part, which we need to know in order to get different things such as scree plot, biplot, and I will also talk about rotated component matrix, which we get by giving the rotation. So let me clear it first. Here, I won't get into the technical jargon or the math which involves calculating principal components. Rather than that, I would like to focus on why we need to do PCA, how we are supposed to do it in R, and where we need to take our decision and all. So if you ask me why, the first and the foremost thing which comes to our mind is dimension reduction. Here, the word dimension refers to variable. If you don't know how, let me explain that with an example of P dataset. Here, this is the data of P. So you can see I have intentionally taken only 7 genotypes in order to keep them clean in graphs. And I have also considered some of the categorical variables. So these are all the categorical variables which will be converted into factors in R. And I have also considered some of the numerical variables which can be quantifiable with the help of numbers. So here I will consider yield per plant, and let me show you how can we interpret this variable with the help of graph. When we have only one variable, we can use an histogram to represent the distribution of the data. Here you can see P725 and EC598820 are the two genotypes for which yield falls between 6 to 8 grams per plant, and rest having only one genotype, as you can see in the bins. We can narrow the bin sizes, but that is not our interest here. If we have two variables, suppose plant height and yield, we can use scatter plot, considering yield in x-axis and height in y-axis. Here you can see B22 on scatter plot. Here in the scatter plot, we can guess the height of B22 is somewhere around 65 centimeter, and the yield is exactly around 4.5 gram per plant. In the next case, what we have to do when we have three variables is we can interpret our results on a display screen with the help of computing power from the processor. So here in the 3D plot, if your eyes won't lock on the z-axis, then you understand how it feels. Later, if you try to find the variables, their values, then your brain starts to overclock. So 3D plots are not the best option when documenting, especially on paper. It is not possible. Just imagine if we have more than three variables. What we have to do? We won't have more than three dimensions practically. And don't think about this theory, it's there only on books. So, what we can do is we can address this problem logically. What we can make is we can add some of the additional identifiers in order to identify the different variables. And the additional identifiers works well with the categorical variables. Here I have considered flower color. So that is identified by the standard petal color. Here you can see that the B22 genotype having height around 65 cm with the yield 4.5 gram per plant is also having purple color flower. And we can also consider one more variable. So here I have considered shape as an additional identifier in order to identify the seed surface. Here you can see that the B22 having 65 cm height and 4.5 gram yield per plant with purple color flowers is also having smooth seed surface, and up to here it's okay. When we try to add a numerical variable, that is the time where graph seems to be tough to interpret. Here you can see I have added an additional variable primary branches with the help of an additional identifier, the size. So here how can you compare to get the value of the primary branches just by looking into the size? So here it is okay because we are having less number of genotypes, and this logic won't help when we are having more number of variables or more number of individuals. So that is where the principal component analysis comes in. So how it is going to reduce the number of variables means, so consider this is a data set with n number of variables. Imagine this as an Excel sheet, and the different colors represent the different columns of an Excel sheet where our variables will be stored. So how principal component analysis works means it will create a new data set with n number of principal components in such a way that the first principal component will be having most amount of information from all the n number of variables when compared to the second principal component. So here the first principal component has 29% of information and the second has 20%. That is how when we address a biplot with the first two principal components, we will address 49% of variability here in this case. And this trend continues across all the principal component in such a way that the last principal component will be having least amount of information. So this is what our script plot represents. And the script plot will be helpful in deciding how many number of principal components to select for future predictions. So if you are interested in theoretical part, check out the links given in the description. So with that, I will get into the practical part. So here the first thing we need to know about is the data and its structure. In simple terms, how we need to take our data in an Excel sheet. Here I have stored my dataset in a folder known as Cotton PCA on the desktop. So this is how our dataset looks like. Here you can see I have considered 139 different genotypes along with 18 different variables. Here I have not considered the replication. So each and every row we have unique genotypes. So the total number of rows is equals to total number of genotypes. In case if you have replication, consider the mean value by taking the averages of replication. So at last we need to save the dataset before importing it into the R. With that, let's get into the R Studio and start coding. Here I would like to give some tips for the beginners. If you are a beginner and don't know anything about R and PCA, don't skip the video here because instead of getting something, the chances of getting into the trouble is more, and R is case sensitive. So don't mess with the cases. Capital R is not equals to small r. If the text is not visible, then please do consider increasing the resolution of the video. Make sure that you will pass comma after each and every argument. After completing a line of code, we need to execute that line by pressing Ctrl Enter or Command Enter. By keeping all those things in your mind, let's import the dataset. So click on Import Dataset and select from Excel. If you are using the R Studio for the first time, you may not get this interface. So give all the permission what it asks, then you will get this interface. So from here, we need to click on Browse and select the dataset. Here I have stored my dataset on the desktop on a folder known as Cotton PCA. So within that, select our data frame. Later, ensure that all the variables are in numbers, which mean doubles. In case if you want to change the name of the data frame, you can change that from here. By default, the first sheet will be selected. If you are having more than one sheet, you can change that from here. And this is what the code which runs behind this graphical user interface in order to import the dataset. So click on Import. Once the data gets imported into the R Studio, you can view that from here. And if you want, you can cross verify also. From the script, if you type the name of the dataset, that is Cotton, then execute, you will get a brief outlook at the dataset in the console. And if you want to see the entire dataset, use the view function. Here, the view starts from capital V. Within that, if we mention our dataset name, that is Cotton, we will see our dataset in a separate tab when we execute that line here in the R Studio itself. And next, the most important step in our data analysis is scaling that data. Here we need to scale our data because one of the stepping stone in our principal component analysis is variance and covariance matrix. As we all know that the covariance is sensitive for scale and we need to scale the data in order to avoid a single variable dominating over other variable. Here I will store my scaled Cotton dataset in an object known as SCD, which in short refers to the same only. Here I am going to use a function known as scale. Within that, we need to mention our dataset name, that is Cotton. Later, we need to select all the rows and columns we need to scale. Here, we need to select only numerical variables because we won't do the PCA for categorical variables or other data. Here in my dataset, as you can see, the genotype and name doesn't have any mathematical importance in the PCA. So we need to take out the first two columns. In order to do that, within big brackets, mention comma. So whatever we write behind comma are rows. Since we need all rows, I won't write anything. In order to take out the first two columns, I will write the code as -1 is to -2. That is from 1 to 2. we need to take out the column. That is why I am mentioning minus. Then if you mention center is equals to true or not, by default it will consider center as true only. So the additional argument center is equals to true doesn't have any practical sense here. For the reference purpose, I am mentioning here. When we execute this line, the scale data will be stored in SCD. So execute SCD in order to get an outlook at the scaled quadrant dataset. This is what our scale data looks like. So from here onwards, whenever I mention the dataset, it will be scaled quadrant data only. And in the next step, I will adjust the option in my favor. Here we need to increase the maximum amount of lines which has to be printed in the console. So in order to change that, use options function. Within that, mention max.print as an argument. Then we need to select the number of lines which has to be printed. Here I will select 10,000 lines. So when we execute this line, the option will be changed to 10,000 lines to be printed in the console. The next option, I will turn off the scientific display. In order to do that, we need to mention siphon is equals to 100. This is the code to turn off the scientific display so that we can avoid the logs and exponential terms printing on the console. And the last option is setting the working directory. So from session, set working directory, click on choose directory. From desktop, select the original folder where our dataset has been stored, that is quadrant PCA. If we click on open, the working directory will be set. Then we can use get working directory function, that is get WD. Within parentheses, don't mention anything. So if we execute this line, we can cross confirm our working directory. So after setting the options right, we need to install some of the visualization packages. And the first and the foremost package we need for visualization is FactoExtra. So use install.packages command. Within the parentheses, mention FactoExtra using double inverted commas. When we execute this line, the package starts to install. As you can see the red beep here in the console. So wait until it stops. Alternatively, we can also install a new package from the CRAN. So click on install and type the package name, that is FactoExtra here. So select the package FactoExtra, and when we click on install, the package will start to install. But I will cancel here because I have already installing it with the help of command. So after the red beep stops, here in the console you can see the package has been successfully installed. Then we need to load the package. So use library function. Within the parentheses, mention the package name, that is FactoExtra. When we execute this line, the package will be loaded into the environment. And the next package will be useful for visualizing the principal components in 3 dimensions. So if you are interested in that, then only you install this package. So within install.packages command, mention PCA3D in double inverted commas. When we execute this line, the package will start to install. Since I already have this package, I will skip this line and load the PCA3D package directly into the environment using library function. And this package only works fine with the Windows operating system. I tried using with Mac, but it had some compatibility issues. With that, let's get into the PCA by using built-in functions. So if you have wondered why there are two different functions built in that is in stats packages, because the way in which they works is different. So among the two functions, let's consider the less popular spectral decomposition function. Here I will store my output in an object known as PCA1, and the function which works on spectral decomposition is printcomp from the stats package. So select this function. Within printcomp function, we need to mention our scaled quadrant dataset, that is SCD. When we execute this line, the output will be there in PCA1. So when we execute PCA1, we will get the standard deviation. So these are the standard deviations. When we square a standard deviation, we will get the variance. Here the variance refers to eigenvalue. In order to get a biplot, we need to use biplot function. Within that, we just need to pass the object where our PCA has been stored. Here PCA1. If we execute this line, we will get the plot in plot span. And from the object where the PCA has been stored, we can access these things. So in order to select, we need to use a dollar symbol. So here dollar is the symbol of select. So from PCA1, I will select the scores. When we execute this line, we will get the scores here in the console. So I won't talk much about this function. I will get into the second one, which is most popular, and that is based on single value decomposition. Here I would like to store the results of PCA in an object known as PCA2. And the function which works on singular value decomposition is PRComp from the stars package. So within parentheses, we need to mention our dataset, that is scaled cotton data. When we execute this line, we will get the output in PCA2. So when we execute PCA2, we will find these results in the console. And here we will get the scores and also the standard deviations. So here also same, if we square the standard deviation, we will get our eigenvalues. So in order to select the scores, which means the individual scores, not the variable scores which are already printed in the console by executing PCA2. So in order to get that from PCA2, the object where our PCA has been stored, we need to use dollar symbol. From that, this drop down menu appears. So from here, if we select X, we will get the individual scores. So when we execute the above line, the scores will appear in the console. You can scroll through them. In order to get the proportion of variance and the cumulative proportion of variance explained by the principal components, we need to use summary function. Within that, we just need to mention the object where our results has been stored. Here PCA2. So if we execute this line, the results will be out in console. In order to get the byplot, use the same byplot function. Within that, we just need to mention the object, here PCA2. So here if you observe the byplots from the older function PRINTCOMP and this function PRComp, they are not same. Why? Because if we compare the scores stored in two different objects of the PCA results, that is PCA1 and PCA2, they will not be the same. Why? Because if the values are same, then the vectors will not be same, that is identified by plus or minus. So don't intermix the results from two different functions. You either use printcomp function or use prcomp function. Don't use a byplot from the prcomp function and the scores of printcomp function. With that, let's move to the visualization part of our PCA. So here our fviz PCA functions from the package FactoExtra comes to use. So here let's see how we can use the FactoExtra function for our visualization. The first function here is fviz underscore PCA. This is the basic function in order to get a byplot. So within this function, we need to mention the object where the PCA results has been stored. Here PCA2. So if we execute this line, we will get the byplot directly. Since these functions provide a high quality, good graphs, they will take a lot of time in order to produce the plot. So wait until the plot appears. Later from here you can zoom in the plot, and if you want you can take the screenshot of this plot from here itself. As you can see, there is a lot of overlapping of labels. So in order to address this issue, we need to use another function, and the function name is fviz underscore PCA underscore byplot, and this function provides us with a lot of customization options. So within that, we just need to mention the object where our result has been stored, that is PCA2. So when we execute this line without any additional argument, we will get the same byplot. So if we pass an additional argument, that is repel, which is equals to true, when we execute this line, we will get a byplot in which the labels are moved aside with the help of a line attached to the point. So here we will get some of the warning messages, and that warning messages is true when we are seeing the plot within the RStudio without expanding it. So when we zoom it in, we will get some of the labels which have been missed. So here you can see, and you can confirm that with your eyes manually. So you can see most of the labels have been there, though there is warning, but some of the labels might be missing. So I will address this issue later, how to get the plot with all labels. And the next function which I'm going to talk is fviz underscore PCA underscore variables. Here the variables have been mentioned in short by var. So within that we need to mention the object where the results have been stored, here PCA2. So if we execute this line, we will get the plot indicating the direction of variables in the byplot. So ignore all those warnings. If you want, you can also use repulsion here. So if you click on zoom, you will get the full plot. As I said earlier, we can also use repulsion here. So pass additional argument repel is equals to true, then we will get a plot like this. So in order to get only information about the individual, we can also use fvis underscore pca underscore individual plot. Here ind is the short form for individuals. So within that, mention the object, it is PCA2, and execute this line in order to get the individual plot where the individuals have been represented on principal component 1 versus principal component 2. So here dimension 1 and dimension 2 refers to principal component 1 and principal component 2 respectively. So in order to get a script plot, use fvis underscore script plot function. Within that, mention the object where PCA has been stored. Here PCA2. When we execute this line, we will get a script plot. I will talk about the customization of this plot later. The main reason, or the only reason, why I used a PRComp function from the stats package is to show you the 3D visualization of principal components. Here, the PCA 3D package comes to our help, and the function which I am going to use is PCA3D from the package PCA3D. Within that, we just need to mention the object of class printcomp or PRComp, that is PCA2. So when we execute this line, the 3D plot will appear in a new tab. So here you can see this is the 3D plot containing three principal components, that is PCA1, 2 and 3. This is only possible with the Windows operating system. I tried using in Mac, but it was not possible. And in order to get help or know more about PCA3D function, type question mark. After that, mention the package name or the function name, that is PCA3D. Here you will get more information. What are the arguments we can use to customize the 3D plots? So here, let me show you the most important customization part, that is adding labels. Here, everything remains same within the PCA3D function, and the only argument we need to pass after the object PCA2 is show.labels, which is equals to true. When we execute this line, you can see a 3D plot with labels. I won't recommend this if you have more number of individuals or genotypes, as the plot is more clumsy. You can only use this when you are having less number of genotypes. And look at all the three dimensions and take a screenshot where you can see more number of individual, and you can use that in your thesis. And we can also make it look a little bit fancier. So in case if you pass another argument fancy, which is also equals to true, then you will get a 3D plot like this. So in case if you are interested in 3D plots, then only you can use this procedure in which we need to run this line of code, and there is no need of PCA 3D package also. So you can directly jump after loading the Facto Extra package. Here I am going to use another package which is very much fine-tuned for our data analysis, and that is FactoMineR. Here Facto starts from capital F and mine starts from capital M and R is capital. So while installing the package, don't get confused with the cases. Use install.packages command within parentheses by using double inverted comma mention FactoMineR as I said above. So there is no need to install this package because I've already installed. I will directly load this package by using library command. So within library function, we need to mention FactoMineR. So when we execute this line, the package gets loaded into our environment. And I'm going to use this package because this package also works on singular value decomposition. Here I am going to store my PCA output in an object known as FPCA. For me, it refers as full PCA since I am going to use all the 18 different components. Here we need to use a function known as PCA. All the letters should be in uppercase. So within that, we need to mention our dataset, that is scaled cotton dataset SCD. Then by default, it only consider 5 principal components. In order to change it, we need to pass additional argument known as NCP, number of components. Here I will select it as 18 because I have 18 different variables. So when we execute, the output will be there in FPCA. So when we execute FPCA, we will see what are all the results stored in FPCA. In order to access a particular part of the result, we need to type this thing in front of our object FPCA. So if we type and execute these things, we will get the results what are stored in that. So let me show you how to get the eigenvalues stored in FPCA. So type FPCA and then type dollar symbol, Then from here we can select the eigenvalue table. So if we execute, we will get the table containing eigenvalues, percentage of variance and cumulative percentage of variance. And this table is very much important for us. I will show later how to import this table directly into the Excel sheet. In the same way, we can also access the different results. So in order to get the scores, select the object, that is FPCA, then use dollar symbol in order to select. Then from here we need to select the individuals because we need individual scores. Then dollar symbol, then select on coordinates. So if we execute, then we will get the scores, the raw principal component scores, and you can scroll down in the console in order to see all the scores. So after that, let's look into the different things which are not so much important yet useful in our data analysis. So the first thing in this part is square cosine. It shows the importance of a principal component for a given observation. In order to find it, select the object, that is FPCA, and use dollar symbol. From here, select the individual, then use dollar symbol. From here, select positive. So when we execute this line, we will get our square cosines. So the next important part is the contribution of variables. So from the object where our PCA has been stored, that is FPCA, select the variables by using dollar symbol. So after selecting the variables, then select contribution, which in short contrib. So when we execute this line, we will get the contribution of different variables. So with that, let's move on to the visualization part. The main reason why I selected the FactoMineR is the compatibility of fviz function of factor extra is better with the FactoMineR package. In order to get a basic biplot, we can use fviz underscore PCA function. Within that, we just need to mention the object here fPCA. When we execute, we will find a biplot. But this is not customizable. In order to get a customizable biplot, we need to use another function known as fviz underscore PCA underscore biplot. Within the parenthesis, mention the object where PCA has been stored, here fPCA. Then the first customization option which I am going to use is repulsion. So mention repulsion is equals to true. If we execute this line, we will find the biplot with the labels moved aside. So ignore the warnings, it will be there. Wait until the plot appears. So when we click on zoom, the missing labels will appear in zoomed version. So here you can see most of the labels are there, but I think some of the labels might be missing here. So we can get this label when we export this graph with adequate resolution. That I will show you shortly. So let's talk about further customization. If you have time to interpret the quality of representation of individual, you can also use square cosine. So here I will use an additional identifier color. So here we need to pass additional argument col.ind, which refers to color of individual. So mention cost2 within double inverted commas. So when we execute this line, we will get a biplot in which we can also determine the quality of representation by color. Since the individuals and variables are having same color, that is blue, I would like to change the color of variables to red. So here in this line only, pass the additional argument col.var, which refers to color of variable. So I will keep it as red. So mention red in double inverted commas. When we execute this line, this beautiful biplot appears. So this is what the biplot which I was looking to export. In order to export, close this window and click on export, then save as image. From this window, we need to select the resolution. Here the resolution will be in pixel. So I will keep the width 1920 pixels. So if we have maintained the aspect ratio, the height will also changes automatically. If you want to change the name, you can change the name of the biplot from here. So if you click here, we will get a preview. So when I click save, the plot will be saved in the working directory and we will get the preview here itself. And you can see all the labels which have been missed in the RStudio starts to appear in the exported image. As I said earlier, this plot will be exported in the working directory. So if we go to the working directory, we will find our plot. With that said, let's move back to the RStudio and start working on code. If you want to know further about customization, you can ask for the help in this RStudio itself. So after question mark, mention fs PCA function. So if you execute this line, you will find help here. With that, let's move toward individual scatter plot. So the first function which I'm going to use is fs underscore PCA underscore ind. Underscore will be there within two words in each and every function. So I won't keep telling underscore underscore in each and every time. So within that function, we need to mention the object where the PCA has been stored. That is FPCA. If we execute this line, we will get the basic scatter plot. Here also we can use the repulsion. So here I will keep repul is equals to true. Here there is no need to mention true with all words. If we mention uppercase T, then R will read as true only. So when we execute this line, we will get the scatter plot with the labels having repulsion. So here also we can represent the quality. So pass the additional argument color of individual and keep it to cos2. So mention cos2 within double inverted comma, then we will get the scatter plot having the quality of representation of individual. With that, let's see what we can get from variables. If we use fs PCA variables function, we will get the plot indicating the direction of variables in first two principal components or first two dimensions. So here also we can keep the repulsion on. So within the fs PCA variable function, mention the first argument, that is fpca, where the PCA has been stored, then pass the additional argument repel equals to true. When we execute this line, we will get the labels having repulsion. And the most often practice what we will do with this variable plot is we will represent the variables by their contribution instead of quality of representation. So in order to get that kind of plot, pass the additional argument color of variables and set it to contribution. So here contrib, that is the short form of contribution, mention that in double inverted comma. So when we execute, we will get this kind of variable plot. And the next kind of plot helps us a lot in determining number of principal components, that is script plot. So here the function which we need to use is fs script plot. Within that we need to mention the object where the PCA has been stored, that is fpca. So if we execute this line, by default we will get only 10 number of components. Why? Because we will select the number of components less than 10 only. That is most often case what we will do. So in order to get all the 18 components, we need to pass the additional argument ncp. Here ncp represents number of components. Since I have 18 variables, I will mention 18. So when we execute this line, we will get script plot with 18 different principal components. Most oftenly, we won't consider the bar in script plot. In order to take out those bars, we need to pass an additional argument, that is geom. Here we need to set it to line. So mention line within double inverted commas. When we execute this line, we'll get a line script plot. So if you want a bar script plot instead of line, within geom mention bar. So if we execute this line, we will get a bar script plot. And if you care a lot about aesthetics, you can also change the color of bar. So in order to change that, pass the additional argument bar fill and set it to red color. So here mention red within double inverted comma. So when we execute this line, we will get a red script bar plot. Instead of using proportion of variance in y-axis, we can also consider eigenvalues. In order to do that, use additional argument choice and set it to eigenvalue. And we can also pass the additional customization arguments here also. And this is what all I wanted to talk about script plot. So the next important part is selecting how many number of principal components to be used for future prediction. And this is highly subjective thing, and there are different way of selecting the number of principal components. You can do either by looking script plot or looking the numbers present in the eigenvalue table, which also contains percentage and cumulative percentage of variation explained. So I will expand the console in order to get a better view. So this is what the table. So in case if you are already fixed your mind, no matter whatever the percentage of variation covered by first three principal components, I will only use first three principal components. That is also good. Here the first three principal component only explain 39% of variation, and You can also target the total cumulative percentage of variation explained by the number of principal components. So if you want 50%, you can select 5 principal components, and in case if you want 90%, you can select 12 principal components. While interpreting our results, we will mention those components having more than 0.5 eigenvalue contains good amount of information about the variability, but we can't use 13 different principal components. What we can do is we can consider those principal components having eigenvalue more than 1. Here 7 principal components. This is what the default method what we will find in SPSS, and here I will consider 7 principal components for my future prediction. The table what you are seeing in the console is very much important for the interpretation of results in the thesis, right? So let me tell you how to import this table directly into the Excel sheet. So click here to collapse the console. So from here I will store the table separately in an object known as table1. So from the object where PCA has been stored, if PCA, select the eigenvalue table by using dollar symbol. So when we execute this line, the table will be there in table1. Before importing, we need to check the class of table1. So within class function, mention table1, and if we execute this line, we will find the class. As you can see, this is a matrix or array. So before importing it into the Excel sheet, we need to convert the matrix into data frame. So select table1 and use as.dataFrame function. Within that, mention table1. So if we execute this line, the table1 will be converted as a data frame. So check the class of the table1 once again, and here you can find this is a data frame. Now this is ready to import into the Excel sheet. In order to import the data set, we need an additional package known as write Excel. So if you don't have, use install.packages command. Within that, mention write Excel in double inverted commas. So if we execute this line, we will get the write Excel package installed in our RStudio. Since I already have this package, I will directly load this package using library command. So within library function, mention write Excel. So if we execute this line, the package will be loaded. In order to import the dataset, we need to use write underscore XLSX function. Within that, we need to mention the table which has to be imported. Here it is table1. After that, we need to mention the name of the table within double inverted commas. So here I will mention eigentable.xlsx. Here .xlsx refers to the format of Excel sheet. Once we execute this line, the table will be stored in our working directory. So if you go back to the working directory, you can find the table here, eigen table. So if we click on this table, we will find the table imported in our working directory. So this is how we can import any table, and you can import any table if you want by following same method. So in other principal component analysis videos, I did see a question: what is the most technical way of judging number of principal components? So in order to address this question, we need to convert the eigenvalue table into a data frame first. Since I already converted the table into a data frame, here I will use plot function to plot the cumulative percentage. So within plot function, select the table 1 and use dollar symbol to select the column. So here select the cumulative percentage of variance. So if we execute this line, we will get this plot, and here we need to look at the point where we will get a straight line. Here we will get the straight line, and this is what the elbow method or branching method denotes. So I need to consider 15 different principal components based on this method. So here I won't consider this method. I usually follow the SPSS default method, and I do consider 7 principal components for the rotation. So this is what all I wanted to tell about principal components. So in case if you have any doubt regarding principal components, attach it in the comment section below. Now I am going to the next concept, that is rotated components. In order to use rotation, the only package which comes close to the SPSS is psych. So we need to install the package psych. In order to install the package, we need to use install.packages command. Within that, mention psych in double inverted commas. Since I already have this package installed, I will load this package directly into the environment using library command. So within library function, mention psych and execute this line to get the package loaded in RStudio. So here don't worry about using a different function from a new package psych in order to give rotation. Why? Because if we give rotation, we do abandon the principal components. That is the reason why I am using a new package psych for rotation. In case if you are not convinced and want to know much more about rotation, check out the article given in the description below. So here I would like to store my results in an object known as RPCA, rotated principal components. And the function which I am going to use is principal. Everything should be in lowercase only. Within that, we need to specify the dataset, that is scale coded data, SCD. After that, we need to specify the number of components has to be rotated. Here it is 7. After that, we need to specify the type of rotation. And the most famous and most often used rotation is varimax. So mention varimax within double inverted commas. And we need to give an additional argument, that is scores. And we need to keep it as true because we need scores for our interpretation. When we execute this line, the output will be stored in RPCA. So when we execute RPCA, we will get our results. So this is what the results looks like. Here don't get confused from the terminologies what we get in the results. You can also consider rotated component as principal component, but that is not appropriate. In older cases, we find principal component instead of rotated component, and we have inherited the same thing from thesis to thesis. What someone followed using some software, which throw the result as principal component instead of rotated component. Here I don't get into that discussion. If you want to know about that, check the article given in the description below, which tells more about the rotated components. So here you can find the results along with the rotated component matrix, that is RC1 to RC7, and here H square refers to the commonalities. And we can also access a particular section of the result. In order to do that, from the object where our rotated components have been stored, that is RPCA, use dollar symbol. And from here we can select a particular thing. So I will select commonalities here. So when we execute this line, we will find the commonalities here. And in the same way, we can also access the loadings of rotated component. In order to do that, from the RPCA, select loadings. When we execute this line, we will get our rotated component matrix with the cutoff. And why this happens? Because in order to find the association of a variable with the rotated component, here you can find that the first variable is strongly associated with sixth rotated component, and the second variable is also associated strongly with the sixth rotated component. And we can also stop this kind of results while printing. So in order to do that, within print function, mention the object where the loadings have been stored, that is RPCA. Within that, select loadings and pass the additional argument. Here I will first get a round figure of 3 digits from the decimal point. Later I will pass an additional argument telling cutoff should be equals to 0. When we execute this line, we will find a normal rotated component matrix here in the console. And this is a little bit tough to get into the Excel sheet. So we need to follow an indirect approach. In order to do that, we need to expand the console first. Then I will take a screenshot of the figure containing the rotated component matrix. Then I will save it in the working directory. After saving it in the directory, I will get into the Google Chrome and search for image to Excel sheet converter. Then we will find this website. From here we need to select extracttable.com. So when we enter this website, this kind of interface will appear. So click on anywhere here. Then this window opens. From this window we need to select the location where our screenshot has been stored. So if we click on open, it asks permission to upload this image. So click on OK. Then the image will convert into an Excel table automatically, as you can see here. Then select the format in which you would like to download this table. I will select Excel and click on download. So once the table has been downloaded, you can enable editing and change the order or the sequence of rotated components. And if you want, you can plot the rotated component matrix here only, but I will do that in the RStudio. So I will collapse the console, and in order to get a rotated component matrix bar plot, we need to use the bar plot function from the graphics package. Within that, if we just mention our loadings stored in RPCA, we will get this kind of bar plot, which is completely basic and that too stacked. And in order to get a bar plot in which the stacking is not there, we need to pass an additional argument, that is beside, and we need to keep it as true. When we execute this line, we will get this kind of bar plot, and it is very much easy to change the color of bars. So in order to do that, we need to pass another argument, color. So COL is the short form, and we need to set it for the desired color. Here I will set it as blue. So mention blue within double inverted commas, and I would also like to add the main heading. In order to do that, use another argument, main. So for main, we need to mention the heading within double inverted commas. So mention the heading here, rotated component matrix within double inverted commas. When we execute this line, we will get this kind of bar plot. For reference purpose, I will bring the main argument to the next line. So hit enter after the comma. So in order to get the fancy plot, what we see in our thesis, in which the bars will be having different colors, which indeed refers the different variables. In order to do that, we need to have an additional or color palette. The only package which can handle 18 different colors in a palette is Pals. So if you don't have this package, install it using install.packages command. So within parentheses, mention PALS using double inverted commas. Since I already have this package, I will directly load this package using library function. And you can just copy this line and paste it over here. So here we need to change the color, right? So take off the blue and use the function which is present in the package pals. So here I will use brewer.accent as the first function. Within that we need to mention the number of variables or the number of colors. So here 18, because I have 18 different variables. So when we execute this line, we will find this kind of bar plot. So you can see there is a little bit of overlapping in these colors, so that is acceptable. And we can also try different shades of a particular color. So here same, I will just copy and paste this line, and I will change the function of color to brewer.greens. Within that we need to mention the number of shades required. Here I need 18 different shades of green, so I will mention 18. When we execute this line, we will find a bar plot like this, in which 18 different shades of green has been present. So you can also play with the different color palettes which are available in the package pals. So at last I would like to show once again how to get this bar plot. We need to use bar plot function. Within that, the first argument will be our loadings. Then we need to avoid the stacking of bars. So pass the argument beside is equals to true. After this argument, we need to select the color. So here I will select the color band. So in order to do that, we need to use alphabet function. There are two functions, alphabet and alphabet 2. You can use either of this, and if you want to know about their colors, I will give that in link in the description below. So within alphabet, we need to mention the number of colors. So mention 18. Then we need to pass the main heading. So mention rotated component matrix within double inverted commas. When we execute this line, we will get this kind of bar plot, which is very much appealing to our eyes. So you can use this method to get the rotated component matrix plot, which I recommend for you when the number of variables is less than 26, that is equals to number of alphabets. So here we can also rearrange these rotated components by using code, but it takes a lot of time. So you can do that manually in a Photoshop or presentation. What I recommend after rearranging these rotated components is the 0 what you see in the scale should align with this line in the bar plots. So that is the correct method to realign. So finally we are in the last part of the video. Here I will show you how to import the scores of rotated components into Excel sheet. So if you don't know where the scores have been stored, they are stored in the object RPCA. So use the dollar symbol to select the scores. When we execute this line, we will find the scores here in the console, but they are very much hectic to read from here. So I will store the scores in an object known as scores. When we check the class of scores, we will find that it is a array or matrix and we need to change it to a data frame. So push the scores into as.dataframe function and change the class from array to data frame. So if we check the class of scores, we will find that it is a data frame now. So use writeXLSX function. Within the parenthesis, mention the data frame name as first argument, and the second argument is scores.xlsx, the name with which we need to import the table along with its format. So if we execute this line, the table will be there in our working directory. So go to the working directory and you will find the scores table. Here, the first thing we need to do is we need to add serial numbers as identifiers. Then you rearrange the rotated components. Then we need to sort the table in either direction. So first sort the table from smallest to largest, then from largest to smallest. Then we need to identify the top 10 genotypes based on their scores. And we need to also identify the genotypes which are having scores more than 1. And we need to do this ignoring plus or minus signs because those are vectors only and they don't have any real value in this scores. So the easiest way of identifying the scores is converting from the normal font to bold font. So hit Ctrl B to mark the scores. So we need to continue the same treatment for all rotated components. Finally, with the help of identifiers or the serial numbers, we can rearrange the sequence of genotypes. So when we sort it by smallest to largest number, we will get the original sequence. And we need to mark the top scores by considering different rotated components in combination. So I do attach the thesis for reference in the description below. So read that, it will help you for your interpretation. I hope you guys like the content and subscribe my channel because that is the only support I ask from my community. And finally, thank you for more videos. Check out the data analysis playlist in my YouTube channel.